Good evening. I'm Professor Jonathan Van Tam, Deputy Chief Medical Officer for England. And many of you may have seen me giving public health announcements from the podium at number 10 Downing Street. But tonight is different. Tonight, I'm speaking to you as a scientist and a doctor, not a government advisor. Welcome to the Royal Institution Christmas Lectures. Tonight, we're going viral. Wherever there's life on this earth, there are also viruses. And there are actually more viruses on the earth than there are stars up in our universe. And to be perfectly truthful, most of them are harmless to us. And some of them play vital hidden roles. For example, recycling nutrients back into the oceans. But some are deadly, and just one infection can lead to a global pandemic. The first respiratory virus pandemic of the 20th century was in 1918, and it was the H1N1 influenza virus. And we think that that killed 50 million people around the world, and that's more than actually were killed by World War I, which had taken place over the 4 preceding years. And at the time, there were no vaccines. All we had was isolation, quarantine, face coverings, and keeping social distance. Does that sound familiar? Well, since then, we've had many crises. We had the HIV pandemic in the 1980s. We've had crises due to Ebola and also to Zika. And now we find ourselves here in the middle of a pandemic caused by the SARS-CoV-2 virus, SARS coronavirus. The disease that comes from it is called COVID-19. And right now we're still battling the latest twist in that story in terms of the Omicron variant. That's the kind of bad news, but the good news is, in the past 2 years, we've had absolutely amazing scientific learning and achievements on an unprecedented scale. For those of you who are going to be scientists, being a scientist is a team game. You can't know it all and do it all on your own. And for that reason, over these 3 lectures that are coming up now, I'm going to be joined by some top scientists from around the world who've each played a vital role in this pandemic and getting it under control. Now, to explain the secrets of viruses and how our immune system fights them, our first expert tonight is somebody who helped develop the AstraZeneca vaccine at Oxford University. Will you please welcome Professor Katie Ewer?
So I'm an immunologist, which means I study how our bodies respond when viruses attack us. And I also develop vaccines that help to train our immune system to learn to fight back. Now, as we've seen over the last 2 years, rapid development of vaccines against COVID-19 around the world, that success has been built on decades of research to understand infection and immunology. But before we can all understand a bit more of that tonight, there's a few things that we all need to know first. What are viruses? How do they work? And how does our immune system fight them off? Well, to start with, let's see just how small viruses are. And what we've got here is a model of a human hair that we're going to enlarge about 4,000 times. So after 3, 3, 2, 1. So in real life, this human hair would be about 80 micrometers across. But when we've blown it up 4,000 times, it's now about 30 centimeters across. And at this scale, we can start to see lots of things on our body in a lot more detail. So for example, here, we've got some little models of human skin cells. And these have been blown up 4,000 times as well, just like our hair. So if we look at that here, you can see that now, on this scale, a human skin cell is nearly the same size as my hand. We've got some other bits over here as well. So if we were to blow up an E. coli bacteria, this is a bacteria that can cause infection in all of us. In real life, E. coli would be about 2 micrometers across. But blown up at this scale, it's like this. It's about this size. But viruses are even smaller than bacteria. And if we were to blow up a SARS-CoV-2 virus, the same amount as we've blown up this hair, they would be about the same size as these poppy seeds. So still really difficult to see. So in order to see viruses in a bit more detail, we're going to have to zoom in even further. Now, to do this, we've had some help from the pupils at Biddenham International School, and they've been incredibly busy crafting these beautiful models for us of different types of viruses so that we can see their shapes in more detail. In fact, we've almost got like a whole virus zoo here. So viruses can have these quite simple geometric shapes, but as we know, their primary purpose is to replicate. And when they do that, it can be really bad for human health. So JVT, do you want to tell us a bit more about some of these viruses?
Yeah, I recognise some of these, and this one is the one I've spent most of my life studying. It's the influenza virus, and this is the one that gave us a pandemic in 2009, the swine flu pandemic. It's also the one that gave us the 1918 pandemic that I talked about at the beginning. And this one will give you a sore throat, a cough, a runny nose, and a fever. Now, this one over here, I'm afraid, Katie, if you get this one, you're going to get diarrhea and vomiting. This one is the norovirus. And this one up here, this is a real nasty. This is the rabies virus, and this one attacks the nervous system, and it also, I'm afraid, can attack the brain. It's really very serious, that one.
So as we've seen here, we've got lots of different viruses. They can create lots of different symptoms in different parts of our bodies, but all of these viruses are built of the same stuff. On their outside, they have a protein coat. But what's on the inside of a virus? So we're going to look at that in a bit more detail. In order to do that, I would like a volunteer from the audience. Here in the red top with the monkey on. Yeah, would you like to come down? So, what's your name?
Emily.
Hello, Emily. What we've got here is a coronavirus, and we want to know what's inside it. Can you think of a way that we might be able to open this virus up? A fuse here, something to light it with? Yeah, should we have a go at lighting it? Would you like to put these on for me? And this is just a clicky lighter like that. So you hold that, and what I'd like you to do is hold it right at the end here, and then we're going to have a countdown, okay? So we're going to light the fuse. Go on, press it really hard. That's it. Great. Now come stand over here with me. 3, 2, 1. Thank you very much. You did a great job there, Emily. Do you want me to give you a glass and back as well? Thank you. You go sit down. So inside the virus is its genetic material, either DNA or, more commonly for viruses, RNA. And on here is the recipe that viruses need to replicate. And it's shown here as the genetic code, which for viruses, for RNA, is written as A, G, C, and U. And as you can see, there are lots and lots of letters written on this piece of paper. It's quite a long roll. So JVT, can I have a hand to unroll this, and we'll just see quite how big this recipe is. You can see it's really long. There we are, right at the end.
So, Katie, how many letters are on the code for this virus?
Does anyone in the audience want to have a guess? Do you want to shout out how many letters they think are on here? 1,000, million? Well, actually, some of you were pretty close. It's about 30,000 letters. Now, that might sound like a lot, but if we were to turn that into digital information and store it as a file, it would be less than 1 megabyte. So these are the simple instructions that the virus needs to be able to replicate. But the thing about viruses, they can't do this by themselves. They need a host to be able to reproduce and survive.
So, Katie, if viruses needs another organism to multiply and survive, are they alive or are they dead? Let's just see what the audience think. First of all, if you think a virus is alive, put your hand up. Okay, that's about 30% or so, I'm guessing. And if you think a virus is dead, put your hand up. Oh, that might be slightly more. And if you're really not sure and you can't decide, put your hand up now. Just a few. Katie, what's the answer?
Well, JVT, I'm going to let you in on a little secret, which is that actually scientists can't decide either. What we know is that viruses are very, very ancient. They've been important to human life for a very long time. We've evolved closely with them, and of course, as we know, they're capable of causing disease in humans. But one thing scientists do agree on is that viruses are on the boundary of life. So, why don't we compare viruses to some organisms that we know really are alive, like us, like humans? How does viral RNA compare to human DNA? Well, I've shrunk this genetic code for the coronavirus onto just one single piece of A4. So all of those 30,000 letters that you saw on the roll that JVT held up are now shrunk down onto this piece of paper. And to get it all on one piece of A4 is absolutely tiny. So now let's see on exactly the same scale what the genetic code of a human being looks like. And to do that, we're going to go to the Human Genome Library. And here it is. It's absolutely enormous. In total, we've got 118 books here, and every book has 1,000 pages. So just to give you an idea, oh my God. If we open one of these up, and I get my little handy cam here, you can see again that we've used exactly the same writing that we used, and again, that writing is really, really tiny. So there are a total of 3 billion letters in the human genome. That's 100,000 times more than we saw in the coronavirus genome. And if we were to turn this into digital information, it would take up about 3 gigabytes as a normal electronic file. So this just shows you exactly how simple viruses are. And their simplicity is part of the key to their success. So how does something so small and so simple take over and make us so sick? Well, in order to understand this in a little bit more detail, we need to go inside the cell. We're now going to imagine that the cells in our body are a factory, and in order to get in, we need to go through this security door. Now, the cells of our body have lots of doors on their surface, and it's a way for different things to get in and out of the cells. But in order for things to get into the cell, they need to use a special key. I'm going to see if this key will open this door. And we're in. And now we're inside the cell factory. Now over here, we have the boss's office. This is like the nucleus of the cell where all the genetic information that our bodies need to produce new cells and proteins is stored. And over here, we've got the cell factory, and this is like the ribosome of the cell, and this is where all the proteins are produced. So if our cells are working properly, what should happen is the instructions go from the boss's office across the factory floor to the cell factory here, and the instructions are given to produce the products that our bodies need to build and repair. For example, we might need a spanner to repair cells, to repair muscles or bones, or we might need enzymes like this that can break down things in our body that are no longer required. And this is how our cells work when everything is normal and we're happy and healthy. But what happens when a virus infects a cell? I would like another volunteer from the audience, please. In the blue T-shirt here, why don't you come down? what's your name?
Tyler.
Hello, Tyler. So, congratulations, you have just volunteered to be a coronavirus particle. So we're going to make you look like a coronavirus particle now. And the first thing we're going to do is give you a protein coat. So this protein coat is on the surface of all viruses, including coronaviruses. Now, the other thing that a virus needs, of course, is its RNA, its genetic information here on the USB stick.
Thank you.
But what else does coronavirus have on its surface that's really important? Shout out if you know. Spike, yes, somebody said spike. You were exactly right. So have you got any spikes on your coat? Yes, you have. Right. So that might come in useful later. But we're inside the cell, and we've got a virus inside the cell. This isn't what we want. Come on, out through that door. Come on. We don't want any viruses inside our cells. Out you go. And we're going to lock our virus outside. So now, of course, we're inside, the virus is outside, so we should all be safe, of course, shouldn't we? Well, maybe our volunteer, our virus, can get in. So, our virus over there, our coronavirus, do you want to see if you can get in through the door? Oh, well done, he's done it. Well done. So that was difficult, Tyler, but you did manage to get in. And how did you manage to open the door?
With the spike protein.
Exactly. So the spike is the key to opening the doors on the cells. But the doors that are opened by the key are specific for the coronavirus, and not all cells have the right door or the receptor on their surface. They need that matching lock. Now, what does the virus do now it's inside the cell? It could go to the boss's office, but what do viruses really want to do? Yeah, they want to take over and replicate. So let's go back over to our cell factory. And our coronavirus is going to insert its own genetic material into the cell factory. Now, let's see what the machine's going to make this time. What's the first thing to come out, Tyler?
Another USB.
Yeah, so we've got some more genetic material.
Another spike protein.
Some more spike proteins, and what else? All right, and now we've got some pieces of the protein coat from the virus. So we're starting to make all the different pieces we need for the virus to make copies of itself. And now we can see, oh, okay, I don't think this is what's meant to happen. Come over here with me, Tyler, I'm not sure what's going on. Come over here. And we've got more and more pieces of that protein being produced by the cell. And our factory is in big trouble. Our cell factory has just exploded. And this is not good for us because all of those pieces of virus have now been released and now go and infect new cells. And what does that mean? We've all just contracted COVID-19. Tyler, thank you so much for your help. You did a great job. I'll take your coat.
Okay, so what viruses do to us depends upon the types of cells that they can actually break into. And that's often determined by the shape of the key they use. And here is a model of the SARS-CoV-2 spike protein, this red piece here. And that is the specific shape that allows it to bind onto this piece here. And the piece I'm holding, the blue piece, that's an ACE2 receptor that sits on the outside of human cells. But those receptors are most common in the lungs and in the airways. And that's why SARS-CoV-2 causes a primarily respiratory infection. Children have fewer of these ACE2 receptors than adults. And it probably explains why the disease is more serious in adults. Viruses can seem like pretty nasty invaders, but we do have an immune system, and that immune system can fight back. Katie.
So our immune system is made up of lots of different organs and cells and proteins, and it's what protects our bodies from infection with bacteria and viruses. The thing about our immune system is that if it's running smoothly, we really don't notice it's there. The first line of defense in our bodies against bacteria and viruses coming in is actually our skin. And the skin forms a physical barrier that stops everything on the outside getting inside our bodies. But of course, there are weak points in that, entrances into our body, like our mouth and our nose. So we have a second line of defense inside our mouth and nose. Does anybody know what that is? Did somebody say snot? That's right. Bring on the snot. Now, I'm going to need another volunteer to help me with this. Does anybody want to come down and help me? In the hat up there. Would you like to come down? Okay, so you're going to stand around the other side for me. Okay, what's your name?
Isaac.
Hi, Isaac. Right. What we're going to do here is use our nose to look at how much snot the average adult makes in a day. So we've got a range of beakers here going from very big down to very small. I want you to try and guess how much snot the average adult makes each day. What do you think?
A big one.
You're going to go for a big one. Okay, so pick the one you think is right, and if you put it underneath our model of the nose and we switch on the snot, we should find out. So let's have another countdown. 3, 2, 1. Oh, it's coming up pretty quickly. Is this going to be the right size? What do we think? It's still going, Isaac. I think you might need another beaker here. Do you want to get ready with another one? Right, swap those over. Excellent. Yeah, that's it. Well done. Okay, this should be enough now. This is our 2 biggest beakers. That's it. Is it stopping? No, quick, get ready with another one. Well done, excellent. Keep going. It's a lot of snot. Well, we might as well do the tiny one now. We've done all the others. Thank you, Isaac. So it turns out that all of them hold the entire adult output of snot for a single day. Isaac, thank you so much. So as we've seen, the average adult produces around 2 litres of snot, or to give it its proper name, mucus, every day. But actually, when we're well, most of it isn't in our nose. It remains in our airways, and it has a really important role there. It's there to trap anything that comes into our bodies and stop it getting into our lungs. And so to have another look at this in a bit more detail, I'm going to need one more volunteer. Who wants to come down and help me? In the golden top there, would you like to come down? What's your name?
Marta.
Marta?
Yeah.
Hi, Marta. Right, so we're going to have a look now at what happens when viruses attack our airways. And we've got a demonstration here of the inside of our airways. And now it's snot-free, so there's no snot in our airways. We've just got the simple lining of our lungs and nose. So I'm going to give you these viruses. We're going to throw these at it and see what happens. Go on. Have another one. Excellent. So now you can see, when there's no snot in our airways, these viruses are able to stick onto our airways really, really well. So let's see what happens if we line our airways with snot. Have another go. Okay, you ready for this? Okay, let's have another go now and see what happens when we try and get virus in. Try again. Oh, yeah, well done. So now what you can see is, where there's snot, it makes it much harder for the virus to bind on. Where there isn't any snot, you can see that they're still able to stick. And snot's absolutely amazing because it contains special antiviral and antibacterial enzymes which can destroy them. Now, I bet you'll never look at snot in the same way again. Thank you very much, Marta, for your help. now we've seen how wonderful Snot is, but our immune systems also have a search and destroy army of cells and proteins. Now, the first part of this you've probably heard of. The first component is antibodies. These are Y-shaped proteins that attach to the surface of the virus, clump them together, and stop the virus from being able to break into our cells. But of course, antibodies can only bind if they have the perfect shape to do so. And of course, as we now know, this is a problem because viruses have all those different shapes that we saw earlier. So, how do our bodies create millions of antibodies that are exactly the right shape to combat a new disease, a new virus that we've never seen before? Well, it all starts with a very simple game of chance. And we're going to play that game now. And I'm going to need everybody in the audience to be a volunteer for this one, okay? So you, in the audience, you are all going to be a special type of white blood cell called B cells. And we have millions of them all over our bodies. If we took all of our B cells in our body and lumped them all together in one big clump, it would be about the size of our brain. And B cells are absolutely amazing because they all have different shaped antibodies on their surface. Now, what I would like you all to do is look underneath your seats. And hopefully, you should all have a little model of an antibody under there. So if you've got your antibody, could you hold it up in the air for me, please? What we want to know is which one of you has the right antibody to attach to our specific virus.
So here is our target virus, and what we need is the antibodies to bind onto it. So we've made a big one here, and we're going to now play a game. Over to you, Katie.
Great. And the shape of this spike protein is going to be exactly the same as the spike protein that we saw the virus use to get into the cell earlier. So what we're going to do is I would like everybody in the audience to stand up, please. And we're going to find out whether anybody here has got the right shape for our spike protein. We're going to start on this side, on the red side. So JVT, what's the first part of the pattern that people need to find?
3 red blocks.
So if you've got 3 red blocks on your antibody model, stay standing up. Okay, so everybody, I think, is still standing up at this point. What's next, JVT?
So next is 2 yellow blocks.
So if you haven't got 2 yellow blocks, sit down. So we've lost a few people now. And what's next?
Next is 1 green block.
So if you haven't got a green, sit down. So what have we got on the blue part of the antibody model?
So now we need 2 blue blocks.
We're down to just a few final competitors.
Ooh.
And our very last piece?
The very last piece. 3 purple blocks.
Okay, so have we got anybody left? Yes. We have a winner here in the middle. Come on down.
Well done.
Excellent. Turn around and face everybody. What's your name?
Sophia.
Right, so what we're going to do now is we're going to take your antibody, come round the side and have a look. We're going to pop it on there and see if it binds perfectly to the spike protein. And yes, it does. So you're the one with the right antibody out of all those B cells. Well done.
So as we've seen, Sophia's got the best antibody to fit onto our spike protein. But of course, just one little antibody like this isn't going to be enough to fight off the virus infection. There's one more step in the process that needs to happen. Now, this is a model of a plasma cell. It's another really important cell within our immune system, and its job is to produce very large quantities of antibodies of the right type to fight off an infection. So if you would like to take your winning antibody piece and put it into the top of the plasma cell here, and then if you pull the lever there and see what happens, pull it forward towards you. Brilliant. And now we've got lots and lots of antibody to fight off our virus. So thank you very much, Sophia. You did a great job. We have one more weapon in our immune system that can stop viruses in our tracks, and these are the killers in our bodies. And this is actually my area of expertise in research, killer T cells. So antibodies are really good at going around our bodies and mopping up free virus. But once the virus gets inside our cells, only T cells can see them. And killer T cells act as assassins, going round our body, finding infected cells and eliminating the virus in a really precise and accurate way. So we've got this amazing video here that shows exactly how this happens in real life. So in the green, you've got a T cell. It goes around the body with these finger-like protrusions on its surface, and it's actually feeling the surface of the host cell, looking for any indication that this cell might be infected with virus. And if it finds that it's infected, it attaches to the surface of the cell, makes holes, and injects enzymes and poisons to kill the cell. This destroys the cell and everything in it, including the virus. The most important thing about our immune system is its incredible ability to generate memory. This means that once it's seen a threat like a virus once, it's incredibly good at remembering how to attack it. And this is the key to vaccines. Vaccines train our immune system to recognize virus and to produce the right antibodies and T cells to fight off the infection. And this is something we're going to explore in the last lecture.
Thanks, Katie. That was a really fantastic description of what viruses are and how they get in and how we fight back. And tell me something, how did you feel when you knew for the first time that the AstraZeneca vaccine was going to make antibodies and help us on this journey we've been on?
I think more than anything, I just felt incredibly relieved that the vaccine we'd spent so long and put so much hard work in to actually worked, because there's never any guarantee when you make a new vaccine that it will work. So it was a big relief.
And tell us, what are you working on now in the lab?
Well, so I've been working on malaria for a very long time. The world desperately needs a new malaria vaccine. And I hope that one of the things that will come out of the COVID pandemic is that we can make vaccines for diseases like malaria as quickly as we've been able to make them for COVID.
That's brilliant. Thank you so much for joining us this evening.
Thanks,
and thank you. So now we know what a virus is, how big it is, how it can get inside, and how we fight back. Let's turn our attention instead to how we know when somebody's been infected with a virus, because that's quite important. Firstly, if we're going to treat them, and maybe also if we're going to keep them away from other people so they don't spread it. So let's talk about diagnostic tests. But really, if you're going to talk about diagnostic tests, then what we need is a virologist. And our next expert this evening can help us with that. It's Professor Ravi Gupta.
Hi, my name's Ravi. I'm a virologist as well as a hospital doctor, and I've spent the last 15 years of my career trying to understand HIV, but also improve treatments worldwide. And most recently, we achieved the world's second cure of HIV in the so-called London patient, or Adam Castillejo. Since then, of course, the coronavirus pandemic hit, and we dropped everything to work on this new disease. And even now, my team is working on the Omicron variant and trying to understand why it's spreading so fast. From my point of view as a doctor, the first thing we need to know when treating HIV is, of course, is the patient infected? And for that, you need a test, of course. Now, designing tests depends on where that virus is sitting or residing or replicating. In the case of HIV, this replicates in white blood cells, as we know. And so we do blood tests. Now, SARS-CoV-2, of course, replicates or infects cells in the respiratory tract, either the lungs or the nose. And for that reason, we've got a giant nose here, and it's a replica of mine, apparently. So we're going to take this apart and show you where SARS-CoV-2 is actually replicating. OK, so hope you can see here that most of the nose is actually inside the head. The part inside, the soft part of the nose, is actually very small. And the virus really is sitting way back there in cells that express the receptor ACE2. And so you can imagine this is not easy to test for with a swab. And I think to demonstrate this, I'm going to need a volunteer. OK, how about you in the first row? Hello. What's your name?
Helena.
Helena. Thanks for joining us, Helena. Now, JVT, please can I have a swab? Don't be afraid, it's quite large, but this is my nose, so it's going to hurt me, not you. Thank you very much. Right. Let's just imagine now that this individual was infected yesterday, okay, by someone else who was positive. We're going to try and do a test at day 1. So let's see if we can find any virus. Would you like to take that for me? And then just put it into that nostril. You're going to be pushing it forward. That's it, pushing it. Right. Ooh, that hurts. Keep going until you feel some resistance. Just twist it round gently. There you go. All right. And then you can take it out. OK. Ooh, what do we have here? Right, there's some virus on there. So that's at day 1. Right. The person at day 1 is probably not feeling too unwell. They're probably going about their daily business, but there's some virus in there, as you can see. What about day 4? So at day 4, the person may be feeling a bit unwell, cough, some fever. Let's test at that time point and see what we find. OK, so same again. Let's try and get more virus out this time. There we go. Oh, there we go. Right. 2 hands, I think. Very good. All the way to the back. So you can remember you need to go quite far and twist it around. OK, let's withdraw that gently. Fantastic. Wonderful. Thank you very much. That's a day 4 sample, and you can see there's quite a lot of virus on that swab. Now, what about by day 10? By now, the person's feeling better, maybe completely well, and may still get a test because, of course, people get sick at different stages. So let's see what happens at day 10. Let's repeat that swab here. OK, big twist, and let's pull that back. You're very good at this. Right. Fantastic. Thank you very much. Right, let's pop that on there. All right. Thank you very much. That was excellent swabbing.
So what you can see from this experiment is that when somebody's infected, virus builds up in the body, and then as they get better, it clears again. And on the day 1 swab, there's a fair bit of virus. But on the day 4 swab, there's a lot of virus, and on the day 10 swab, there's not very much at all. And this is important in 2 ways. First of all, it's important in terms of how the diagnostic tests perform, and also as doctors, when we get a specimen from a patient and it's positive, we also need to think about when that specimen was taken compared with when the patient first got ill with symptoms, because it might change the result. You can see that.
OK, so we have a situation where on day 1, you may not have very much virus present, but you really need to know if that person's infected because they would then isolate and stop spreading this virus. So it's really important to have an incredibly sensitive test. So the gold standard that we have at the moment is something called PCR. Who's heard of PCR? Fantastic. Right. It stands for polymerase chain reaction. And this has been around for more than 40 years. And the secret of this is the fact that it is trying to identify bits of the genetic code of the virus. It's not looking for the spike protein or any other bits of structure of the virus. It's looking for the code. What we're going to look for when we're testing is a short signature. A signature that's only a few bases long, something like a genetic fingerprint. So we know what the sequence is going to be of that fingerprint, but we need to find a way of fishing that out of a sample. And the sample is in this bucket. The genetic material from viruses and bacteria from our nose are all in there together. We need to find a way of fishing out SARS-CoV-2, but nothing else. And that requires us to understand another concept, which is base pairing. Now, base pairing is something that we have in our genetic code, and that's how we get double-stranded DNA or the DNA helix. And I'll show you how that works, because up here, looking at our target sequence of SARS-CoV-2, we can add complementary bases. So the A pairs with a T, and for example, a G pairs with a C. Okay, so based on that fact, we can design these things called primers. And primers are really just types of bait in a way. We're using our knowledge of the target sequence to design something that will help us pull that out. So what we're going to do now is use this primer, okay, to fish out SARS-CoV-2 and nothing else. And so this should recognize a complementary sequence in that bucket. So I'm going to have to untangle a few things here. It is Christmas, so there's some tinsel there. Right. So as you can see, using my primer, I have been able to fish out a sequence, and it looks suspiciously like the SARS-CoV-2 sequence that we have on the board. So let's pop this here, and you can see that, hey presto, we have pulled out from a mixed bag of different pieces of genetic material a SARS-CoV-2 specific sequence. And, of course, it comes with the primer attached, because that's how we fished it out. So that's the first step. But this really hasn't helped us, has it? All we've done is fish something out that we can't see. There's only one molecule there, and we need to make many, many more in order to be able to detect it with modern technology. So we're going to have to do something. And this comes back to the polymerase that I mentioned earlier. So I think we're going to need a volunteer for this. Let's go to this side in the red jumper. Come forward, please. Right, what's your name? Alexandra. Alexandra, everybody. OK, right, you're going to become an enzyme. You're a protein, like a little robot in a cell. You're going to add little letters or shapes onto this strand so that they match up, all right? So what I'm going to ask you to do is just find shapes that match the ones that you've got here and just pop them onto the Velcro. OK, let's see you do the first one. OK, excellent. As Alexandra does this, I'm just going to explain the type of polymerase we use, because we don't just use a standard human one. We use a polymerase that's derived from special bacteria that live in really high temperatures and that were first isolated in Yellowstone National Park from a bacterium called Thermus aquaticus. And the name of the polymerase is therefore TAC. Now, in order to make more of this, we're going to have to find a way of putting our primers back on, because remember, the primers were the reason this worked in the first place. Now, there's nowhere to put those primers on at the moment, is there? So what we're going to do is break those two apart. And the way we do that is with heat. So can we have some heat, please? It's going to take a temperature of something like 95 degrees in order to break the hydrogen bonds between these two strands. And once we reach 95 degrees, we're going to be able to separate these two. As you can see here, you've now got two strands, and lo and behold, the primers can attach. But before that happens, we need to drop it down to 60 degrees, please, because otherwise these primers won't bind. So we're down to 60. Let's put the primers back on, as we can see here. Right, now you've got the ability for our polymerase friend here to add more bases. So would you like to add some more on? So it's amazing that a polymerase from a bacterium in the middle of a hot spring in the middle of the US now underpins the great majority of our molecular biology, and a lot of our scientific advances would not have been possible without polymerases. And I cannot stress that enough to you. It's an incredible achievement. All right. Thanks very much for that. You were wonderful. All right. Bye-bye. OK, folks. So we started with one strand, and we ended up with 4 here after a cycle of PCR. The number of strands will double each time. So if we were to heat things up again to 95 and cool down again, we would get 8 strands. And then after another round, we get 16, and then 32. Can anybody tell me how many we would have after 30 rounds of this PCR cycling? You can shout out.
A lot.
A lot. That's correct. 1 million. 1 billion. 500 million. It's about 1 billion. OK, so you'd get 1 billion copies after 30 rounds of PCR that would take you around 3 hours. That's an incredible amplification. And it's quite easy to detect 1 billion particles. And that explains to you why PCR is so sensitive, because it takes a tiny amount, makes lots of it, and then you can detect it.
Thanks, Ravi. So now you can see that, use of PCR in the COVID pandemic has been an absolute revolution to us. But it's an incredible technique that we're now using to diagnose lots of other diseases, including TB, HIV. We can use PCR at the scenes of crimes to detect what's gone on. We can use it to actually tissue type for organ transplants. And PCR is becoming quicker and cheaper as the years go by. But as you saw from Ravi's demonstration, it's still quite complicated, and it still takes 3 hours to make those 1 billion copies. It's not the quickest thing in the world. What if we need to diagnose people faster than PCR, and maybe in a simpler way? Can you think of another test that works for that? Give it a shout out. Lateral flow. What's a lateral flow? Ah, yes, all right. Ravi, lateral flows.
Okay. All right, so who's taken a lateral flow? You all know what it is? Right, of course, everybody knows what it is because you had one to come in here, didn't you? Okay, right. So here, to demonstrate this, is a giant-sized lateral flow device generated by our colleagues here at the RI, and it even has a barcode that works, and it has a test strip that's with a C on it that means control, and this means that the test has worked once you put the sample through at the top. And the T, this goes positive, it means that there's coronavirus in the sample. So I think we're going to need a volunteer for this. Someone in the middle now. I'm trying to stay away from the red jumpers. Okay, red jumper in the middle. Come over the side, right. What's your name?
Max.
Max. Max, thanks for coming down. Right, you're going to demonstrate this giant lateral flow test to everybody. Right, so what I'm going to do is give you a sample here, and I'm going to ask you to pour this into the sample well here. Normally this would be liquid, but it's balls at this time. Okay, right, and we're going to count down from 3. 3, 2, 1, go. So as you can see, the control line has come on, but the test line hasn't, and this shows that the test has worked, but there doesn't seem to be any virus in there. So this is a negative test. Right, Max, now we're going to give you a second sample, okay? You did really well the first time. Let's see how your luck is running tonight. Let's pour that down through the sample well. Go. Excellent. Thank you very much, Max. As you can see, this is now a positive test result because the test has worked and you have the red lights coming on. In other words, the test is positive. So this is a sample that had coronavirus in it. Thanks very much for coming down. Okay, so we've seen a positive test result, but how does this thing actually work? Let's look inside. Imagine these balls representing virus particles. They're covered in spike protein, aren't they? They're going to pass through the test. Now, on this red line, in a real lateral flow device, we have immobilized or placed antibodies to spike. And you saw how those were generated and how specific and tightly they bound in Katie's talk. So those antibodies are placed along there, and as the spike goes through, it will bind to the antibody and trigger a color change. And that's how the lateral test works in reality.
Ravi, that was fantastic, and I've always wanted to know what was inside a lateral flow device, and finally you've explained it to me. So, as you can see, we've got one test that's super duper sensitive, but takes a long while, and we've got the lateral flow tests which aren't quite as sensitive, but are really quick and really practical. And that's one of the things in medicine, and indeed in controlling this coronavirus pandemic. You have to choose the tests that fit the purpose. And lateral flow tests, we all know, we've all used them, they're very quick and simple, but the really accurate ones are the PCR tests. And perhaps that now explains to you why the advice is that if you have a positive lateral flow test, you still need to go and get a PCR.
So is there a way of getting the best of both worlds? Something that's quick, like lateral flow tests, and something that's as sensitive as PCR. Now, early in the COVID pandemic, our hospital was overrun by hundreds of patients with symptoms, and we needed a really quick test that could be done near the patients to give a really quick result that was accurate, because if we got it wrong, people could be transmitting coronavirus to each other within hospital, which is a really dangerous scenario. So there was immense pressure to find such a system. And during my HIV work, I had used a test developed by an inventor, and here to explain this test is the inventor, Dr. Helen Lee. Helen, welcome.
Thank you.
Could you explain to us what the original purpose of the machine was?
Well, originally, this machine was to test for HIV in Africa. in the villages, and we had to check whether the HIV treatment was working, and also whether the babies that are born to HIV positive mother is in fact infected or not. And we take the test to the patient rather than the other way around.
So Helen, can you explain to us how this works?
Okay, it's very simple. I'm sure you have all done a lateral flow test, but I've seen this before, right? A tube with a swab in, yes? So what I've done is that I've transferred with a pipette 300 microliter exactly into this sample tube. So you can see I have this cartridge, I put it in here, and then the second cartridge, I put it here, and the third one, I put it here, but you can see I cannot put it in the wrong way. And now I'm going to put the detection cartridge in. And this, in the NHS sites, some of them call it the ballerina because it dances and twists, and you see. So then I put this sample here. Now I'm ready. All I had to do is to load it. It's simple. Okay, so now I just close the lid. And then what happens is that this particular tablet will prompt me to do the next thing. So now I just say start. It's a combination of PCR and lateral flow. It amplifies like PCR, but at a constant temperature. And so the target also gets to be amplified to billions of copies. But then the detection is visual, just like a lateral flow. If it's negative, it has only one line that shows that the chemistry worked. And if it's positive, then you have two more lines showing that you have COVID.
And can you tell us what the benefits are then of this machine?
Well, the main thing is that it is accurate, it's rapid, it gives you the results in about an hour.
And can it be used for any other viruses or other diseases?
Yes, we just developed a multiplex test which detect the COVID plus flu A, flu B. And so it's a multiplex test. It's going to be shortly in the hospitals, in the care homes, and even in some schools.
Fantastic. So that's incredible. You've got a test that can do multiple different viruses and tell you fairly quickly what the diagnosis is. And that's incredible for a machine that was developed initially to test for HIV in resource-limited settings where there was poor access to PCR. Thank you very much.
Ravi, what a tour de force on diagnostics. If I asked you 2 years ago whether you thought we could get to where we've got to now with diagnostics for coronavirus, what would you have said to me?
Well, I wouldn't have believed you because, of course, to bring tests from development all the way through to clinical use takes a very, very long time. A lot of validation, a lot of studies. And so the rapidity of this has just been incredible. I'm really stunned at how many different tests we have of high-level accuracy.
Thank you so much, Ravi. Thanks for being with us this evening.
Thanks very much.
We've seen that rapid testing can increase the quality of patient care, but could there be even faster ways of testing? Let's bring on our next set of guests, Mark and Scott, and Jodie and Millie. What we've got here is we've got 4 Christmas stockings with 4 different things. Actually, the things are on the backs, on the little sensor panels. And in 2 of these Christmas stockings, we've got old sweaty socks. But one of the old sweaty socks was worn by somebody who had COVID-19. There's no COVID-19 around this evening, please don't be afraid. But the smell of COVID-19, have you ever smelt COVID-19? Might still be on one of the socks. And on the other 2 specimens, we've got wound specimens, and one of those wound specimens is infected with a nasty bacteria called Pseudomonas. And Pseudomonas can be quite serious for patients who've got damaged immune systems. And the other swab has got nothing on it. It's not infected. And so what we're going to do is see if our little friends here can actually find these infections for us. So let's start with COVID. Which dog are we got COVID?
This is Millie.
So Millie, this is Millie. Millie's been specially trained to sniff out COVID. Over to you, Mark.
Ready? 6.
She's found something.
Yes, good girl.
Very good.
Good girl.
But was she right? Yes, she was.
Jodie.
Well done. Right, Jodie, you've got an act to follow now.
Good.
It's your go. Jodie's looking for Pseudomonas.
Ready? 6. Yay, good girl. Good.
Oh yes, she's found it as well. So there you have it. Dogs that have been trained to sniff out infectious diseases. Now I hope we are about to be able to be joined by Dr. Claire Guest online, and here she is. Good evening, Claire. Can you hear me okay?
I can indeed. Good evening, everyone.
Thanks for joining us. Now, you know a lot more about using dogs in medical detection than I do, so I'm just going to ask you the questions. How could these dogs be used in the future?
Well, what you've just seen is a biosensor with a fluffy coat and a waggy tail. And as we know, dogs have been used for many years keeping us safe, finding drugs and explosives. And what the dogs have done here is found the odour of disease. So when we have a particular disease or condition, our odour changes, and the dogs, with their incredible sense of smell, can be trained to detect this and warn us.
So we can't smell these diseases, but the dogs can.
Absolutely. They've got 350 million sensory receptors dedicated to olfaction. Now, I don't know if anybody would like to guess, but us humans have got 5 million, which means we can smell a teaspoon of sugar in a cup of tea. These dogs can smell a teaspoon of sugar in the volume of water held in 2 Olympic-sized swimming pools. Quite incredible sense of smell, and the dogs are really happy to put this nose to work to find disease. For example, Millie, the COVID dog, she could go out now. She could have screened every person as you came this afternoon. So these dogs, because they're very rapid, they could screen hundreds of people at a time.
Thank you, Claire. Thanks for joining us this evening. And thanks to Jodie and Millie, and thanks to Mark and Scott. So I think you've seen in this lecture just a first glimpse of how the pandemic has really been transformed by scientific achievements. And in the next lecture, we're going to think about how these viruses spread, why some humans spread them better than others, and what we can do to stop them. But that's for next time. Thank you for being such a great audience. I hope you've had a good first lecture. Thanks for joining us.